Okay, welcome everybody to another seminar of GEOTOP-A, and it is our great pleasure to have Raymond Goldstein to give a talk on the geometry of multicellular life. To you, Ray.
Thank you very much, Renzo, and thank you to the organizers for the opportunity to speak here. It's a very interesting seminar series you have, and I'm pleased to be a contributor to it. So when the organizers asked me to speak, I was unsure of what would be the best topic, but then I thought you might be interested in hearing a story about some work that sits at the intersection of physics, mathematics, and biology, and it has to do with an important problem in biology. And maybe I can start by saying to you that if I asked you to name some of the fundamental problems in the field of biology, the outstanding order one problems out there, you'd probably first say something along the lines of the origin of life. And then perhaps the second kind of example you would give would be maybe the nature of consciousness. What does it mean to be sentient and aware of ourselves? But actually, there's another problem, which is almost up there in terms of its visibility in the social world, but is talked about a little bit less, which is how it was and why it was that the first organisms that first appeared on life on Earth out of the primordial soup, and which in some ways probably resemble bacteria, eventually evolved to become as complex as we are, with trillions of cells and many different cell types. And this question of the origin of multicellularity is something that has been of interest in science for, well, the better part of a century in a formal sense. And I think it's an opportunity to address a number of interesting questions at this intersection that involve both geometry and topology. And if you think about the simplest forms of life, the simplest uni and multicellular forms of life, the kinds of issues that they must contend with are often physical ones, like buoyancy and diffusion and mixing, because the exchange of nutrients with your environment is one of the most important features of life. So it's not surprising that many physicists and biologists and mathematicians are drawn to this subject because of those natural connections. So that's what I want to talk about today, some geometrical aspects of multicellular life. But I hope you'll sort of forgive me if I also lay on top of this an interesting little story that is something that is also present very much at this intersection between the three disciplines, which is a question about whether research actually requires a hypothesis or not. And as I go along, I will explain what I mean by this and hope, especially for the younger people in the audience who might be involved in interdisciplinary research, you may find this interesting to see the differing viewpoints there are out there as to how to decide on a research direction. Okay, so whenever you're confronted with these kinds of order one problems in biology, it's useful to start looking at some data to get an idea of what you're actually trying to explain. And the data that I show here on this first slide is some of the most interesting data that I've ever seen in this area. It was first presented to the world in an article by Bell and Moores in 1997, and then it was sort of reinterpreted and highlighted by the late great biologist John Bonner, who spent much of his 90 some odd years at Princeton thinking about the origins of multicellularity. So it's a collection of data that shows on the x-axis the total number of cells in organisms of very many types, plants, animals, fungi, seaweeds, ranging from 1 all the way up to 10 to the 15. And on the y-axis, it's the number of distinct types of cells found in those organisms. And again, it's a much smaller range, maybe 1 to 100 or so, but we can start to get a sense of what's going on by looking at this graph. And of course, if you're comes from a statistical physics background and you're used to log-log plots, you might think, there's a straight line somewhere here that I could draw. But I would caution you not to do that, partly because it's obviously very noisy, and I'm not sure what statistically meaningful slope you could assign to this. But secondarily, independent of whether there's a nice exponent, there clearly is a general trend that as organisms get larger in the sense of total number of cells, they get more complex in the total number of cell types. And just for reference, a bacterium sits in the lower left-hand corner, one cell type. And by cell types, you can think, for instance, in the most simple case of performing the functions of life like metabolism, photosynthesis, things like that, and also reproduction. So sort of metabolism and reproduction are the main two players in cellular life. And a human sits way up on the upper right. We have about a 10 to the 14th cells in our body, and by the method of counting in this paper, something on the order of 210 distinct cell types. Now, rather than confronting this entire range of complexity, many scientists of both biological and physical origin have thought for the better part of a century that it's good to be reductionist and actually to focus, for instance, on the lower left-hand corner of this graph and to try to understand a very simply stated question. As I move from one cell in an organism to 10 or 100 or 1,000, and I start to see the appearance of not just one cell type, but two or three, that if I could understand those elementary transitions, I might begin to be able to understand the whole range of what's going on here. And if we can't describe something as simple as the transition from one cell type to two, we're probably not going to understand the entire spectrum here. So when it comes to this kind of well-defined question in biology, it's natural to search for the right organisms to study to make sense out of it. Now, for those who may not know, I sit in the applied math department here in Cambridge, but I also have a working cell biology lab. So we do experiments and theory in trying to answer these questions. And for many years, we have gravitated to a set of organisms that are green algae. You can find them at the River Cam. By just putting your hands in the water and scooping it up, you'll get a number of interesting green algae. And these are well known as model organisms for this transition problem to multicellularity. And what you see here is a montage of pictures of a whole bunch of distinct species, starting on the left with an organism called Chlamydomonas, sometimes called clammy to its friends. It's a green alga, photosynthetic, about 10 microns across. Remember, if you have any hair, a typical hair is about 100 microns across, so this is a tenth of that. It has these two flagella that you can see here with which it swims through its fluid environment. And it turns out that there are these distinct species that are basically built out of 2 to the n, where n is an integer, of these chlamydomonas-like cells in some kind of geometric arrangement. So there's a species called Tetrabena, which is basically a flat plate of 4 of them. There's gonium that is 8 or 16 cells, also pretty much in a flat plate. And then you end up with larger species, 32, 64, 128, all the way up to cells that can, as organisms that can have 50,000 cells in round number and be several millimeters across. So this spectrum of species covers microns to millimeters. So that's a huge range of sizes and an even larger range of cell number. But the most interesting thing to point out first is that when the organisms get beyond the simple gonium-like structure, they tend to be spherical, and the cells are actually mounted in a transparent polymeric matrix called an extracellular matrix. And you can see that very well by the time you get to an organism like Volvox, which is 0.5 millimeter across. And in Volvox, but not in all the other species below, there are actually 2 cell types. Every cell in the smaller species does all the functions of life, which means photosynthesis, let's say, and reproduction. But in Volvox, the reproductive capabilities are sequestered in a small number of cells on the inside of this sphere, and the cells on the outside are essentially the galley slaves that just keep swimming and don't do any reproduction. So there you have it. Pleiodorina has 1 cell type, and Volvox has 2. So there's a sudden transition to 2 cell types. And the fundamental question is, why did nature put the dividing line there? What is it about the size or the number of cells that makes it evolutionarily advantageous to have 2 cell types? So that's an important overlying question in what I'm going to talk about. Now, here's a beautiful video of Volvox in its native behavior that I took in helping the BBC with a documentary some years ago about microorganisms in fresh water. So here you see it tends to spin around an axis as it swims. It's a beautiful geometric structure. The coordinated action of all of those flagella leads to this combination of translation and rotation that is very important in its life cycle. And people have been captivated by this ever since Van Leeuwenhoek, the perfecter of the microscope, wrote a famous letter to the Royal Society in 1700, where he announced the discovery of this wonderful organism. But if we look at a bit more modern times, there's a very interesting book which I commend to you all to read, written by Julian Huxley, one of the many Huxleys in British academic life, called The Individual in the Animal Kingdom. And it's got a beautiful drawing of Volvox at the beginning. hand drawing, obviously from a microscope view, incredibly detailed, showing these daughter colonies growing up inside and all the other cells. But one of the things that Huxley was trying to understand was an elementary question about evolution. So I know this is a mathematics seminar, but I'm going to have a lot of biology today. I hope that's okay. So the question that was being asked is, on what unit of life does natural selection act? Is it on the whole organism? Is it on the individual cell within the organism? How do we figure this out? And this whole book is a kind of explanation of this question and the kinds of model organisms that one could imagine useful to study. And Volvox featured prominently in this book. And in fact, when Huxley talks about different methods by which cells could come together to form multicellular structures, he wrote the following: it was thus the other method, aggregation of similar units and subsequent division of labor among them, that opened to life the full resources of the second grade of individuality of this organism with many parts. In some colony like Volvox, there once lay hidden the secret of the body and the mind of man. Eloquent justification. I feel fine studying this organism as a model. Okay, so my last point of introduction here is to emphasize to you that these green algae of this particular lineage have turned out over the last 20 years or so to be tailor-made for the study of many interesting problems, not only in biology, but also in biological fluid dynamics. I wrote a review article, well, 10 years ago now, where I tried to explain all these different features. So on the upper left, you can study issues of multicellularity, like I've been talking about. The next panel over shows a movie of these spinning organisms that happen to have swum up to the top of a glass chamber, and we're looking down at them. And they spin around as they swim, and they attract each other hydrodynamically and form what we call a hydrodynamic bound state, which is a very interesting problem that has become very important recently in the study of so-called non-reciprocal interactions. The organisms are very large and swim in a very stately manner, so it's possible to use high-resolution techniques in fluid mechanics to map out the flow fields all around them, the streamlines you see here around Volvox, and to connect that, theoretically, to the action of the thousands of flagella that are actually pushing the fluid around on the surface. There are also interesting problems involving tracer particles kicked around by the swimming flow field set up by all these organisms and the existence of non-Gaussian statistics, so a kind of deviation from what you'd expect in ordinary Brownian motion. You can also look at the synchronization of the two flagella in single organisms or multiple flagella in larger ones that are very important in the way these cilia in your respiratory system function, because the flagella of these organisms is essentially identical to the cilia in you. And you can also study questions like, how do organisms that have no central nervous system actually steer through their environment? As you see here, they can actually do what is called phototaxis. They steer to the light because they're photosynthetic. How do they do that? And more recently, as you see here, these organisms have become interesting to physicists because they execute various kinds of tissue folding processes that are important in development, that are very reminiscent of things that we find in us. So there's a lot to study with these green algae. Okay, so the starting point of what I want to talk about today is a very interesting idea that was put forward by two biologists, Thibaut Brunet and Nicole King. Thibaut was a postdoc with Nicole in Berkeley. Thibaut's now at the Institut Pasteur in Paris. He wrote an interesting article about the origins of animal multicellularity 8 years ago. And in it, they talked about three different kinds of organisms, which, in their view, actually have some interesting connections. The first are called choanoflagellates. So these are unicellular and multicellular organisms that are the closest living relatives of animals. So if you think of the tree of life, there's the animal branch, and at the base of that branch, the simplest animals are sponges, with some debate, but basically sponges. And just off to the left or right are the non-animals that are close relatives of those. And these are, the living ones are called choanoflagellates. And it turns out as these organisms grow and divide, they typically form interesting geometric structures that often resemble a rosette geometry, as you see here, with the cells all radially oriented. But they're held together with an extracellular matrix, like I mentioned before, a polymeric cross-linked network of glue that holds them all together in this beautiful form. It turns out that sponges, especially when they're young and have an embryonic development, go through shape transitions that are very similar to the kinds of things that you see, like I just showed you in Volvox, where they can change their geometry and topology. And finally, in the Volvocales, as I've been describing, the generation through reproduction of the multicellular form is maintained by this extracellular matrix that holds everything together. So in all these organisms, it turns out there's an extracellular matrix, which is polymeric material secreted by the cells that organizes the structure into some kind of regular geometry. And the more I thought about this, the more I realized there was an interesting and poorly studied question in biology that was, I thought, worthy of study. And this is how I framed it. How do cells robustly make structures external to themselves? So if you think about it, these very simple cells are excreting materials, building blocks of this matrix. And they don't have any arms to control this. They don't have any eyes to see what's going on. And yet it self-organizes in some very regular geometric structure. And we've seen it with algae, we've seen it with choanoflagellates, and we've seen it with sponges. So how do they do this? How does the inevitable level of fluctuations at the scale of individual cells disappear by the time you deal with a large organism like Volvox that's this beautiful round sphere and grows as a sphere? So you can see it on the scale of tens of microns or hundreds of microns in algae and choanoflagellates. And if you look at the typical cross-sectional view of a sponge, not a kitchen sponge, but a marine sponge, in the most highly developed form, it turns out there's a myriad of spherical chambers that you see shown here that are lined with filter-feeding cells that are like those choanoflagellates, all embedded in an extracellular matrix. And interestingly, a sponge starts life as a sphere, so it's got a genus, not its biological genus, but its mathematical genus of 0. By the time it becomes this porous microfluidic network when it's an adult, it has a genus of, I don't know, 12,009. And so how does it do that? How does it reproduce this microfluidic network in such a way as to create this incredibly porous structure? So I actually interviewed for a grant proposal some years ago in London, and I was presenting this question as an interesting biological question. And I said, well, the proposal is we're going to measure, quantify the geometry of these different organism types as they grow, and from that try to understand the rules that underlie the development of regular structures. And the chair of the panel, who was a very eminent geneticist, said to me, but what's your hypothesis about how this happens? And I said, well, I don't have a hypothesis yet because we haven't done the experiments. I will do the experiments, and then I hope they will motivate me to come up with a reasonable hypothesis about what's going on. And she looked me in the eye and said, how do you know what to look for if you don't have a hypothesis? At which point I knew I wasn't going to get the grant because I'm usually pretty good at answering these questions, but I was totally speechless because I hadn't encountered this kind of pushback against the idea that you do experiments and then you formulate a hypothesis. So it was very disturbing. And I went home and I looked up the definition of the scientific method. There isn't a Cambridge English dictionary, but there is an Oxford English dictionary. So I settled for that. And it says, the scientific method is a process consisting of systematic observation, measurement, and experimentation, formulation of hypothesis, and then deductions and refinement of that. So it's a kind of circle. And I always kind of imagined you started the experiments and then you kind of go around. But apparently that's not true. Apparently you're supposed to start with the hypothesis, at least according to some people, and then proceed from there. So this was kind of disturbing. It's part of a general tension that I have found between the biology world and the physics and math world about the way to do science. And another one of those that I actually wrote about some years ago in eLife, a prominent biology journal, is the question of whether mathematical and theoretical results, that I'm sure most of the people in this audience would consider a result, is actually a result in quotes in the sense of a biology paper. Do you put it in the results or do you somehow put it in the discussion? Is it really a result? And this allowed me to write the world's shortest abstract. Yes, I think they're results. And I'm proud to say that this paper has been viewed 27,000 times, but I don't know if it's had any impact. That's a separate matter. So probably most people just disagree with it. But anyway, let's keep going. So the first part of my detailed talk is about the cellular neighborhoods in Volvox. So here again, I show you this beautiful picture by Huxley, and you can see that each of the individual cells is surrounded by some sort of polygonal network that most of the time looks like a hexagon. And if you take a modern view of this, like you see here in these beautiful images by Moreno Gill, and you look carefully at a Volvox colony using a technique called differential interference contrast microscopy, you can see there actually is really a physical um, compartment surrounding each of the cells in the Volvox. So these are each of the 50,000 cells or so, or 1,000 in this species. And there's a kind of denser region of this extracellular matrix that shows up this polygon very nicely. And these are essentially compartments within the extracellular matrix. You can think of it literally as the neighborhood of every cell. And if you um, uh, use some modern image processing methods, as my brilliant student Anand Srinivasan has done, you can identify all those features and actually show what the neighborhood looks like in some way that allows quantification. And first of all, you can see that most of the polygons are hexagons, but there are occasional pentagons, like you see here. But actually, there's quite a distribution of sizes in the sense of, let's say, areas of these different domains. So it's not every cell is the same if you think of what you see as the output of each cell. So this really raises the question. There's stochasticity at the level of individual cells in terms of the amount of material they spew out. But somehow there's regularity on the scale of the entire organism. And it's that connection that I would like to understand.
Ray,
Yes.
I have a very basic question on the picture you showed just now. Suppose we look at these, the profile of these pentagons or hexagon. What they would, what, what we, what is the interpretation of these profiles? They
Uh,
represent what? I understand this kind of confinement, but the boundary
Yeah. So
represents
what I
what?
will say more about this in a few slides, but
Okay.
what you should think of is that this polymeric network that is self-assembled outside the organism has structure to it. So there are parts of it
Mm-hmm.
like the part that's right around each cell that are a bit more gel-like, and there's a more rigid, denser region that defines the boundary around each cell. In
Mm-hmm.
some ways, I will show you a little bit that this is kind of like a foam, where
Mm.
each cell
Okay.
is secreting a kind of polymeric front, and they collide
Sure.
together and
Sure.
form this structure. Yeah.
Okay.
Okay.
Sorry. Thank you.
No problem. So in fact, if you look, this extracellular matrix is actually a complicated three-dimensional structure. So if you look in cross-section, here's a stylized view. Here's each individual cell. Here are the two flagella that come out. They sort of tunnel through the outer layer, which is called the boundary zone. And here's this compartment around each cell. But then there are layers inside. So the daughter colonies grow up. They're called gonidia when they're undifferentiated and undivided. They grow up inside a vesicle, which is basically more of this extracellular matrix with a fluidic interior. And there are other layers as well. So it's an amazing self-assembled process. And there's no understanding at all of how on earth this geometry emerges from some kind of, I don't know, reaction diffusion system of polymerization, basically. So that's the kind of order one question that's out there. But we can nibble at that question by starting to ask about what's going on with these individual compartments. Okay, so one thing I should tell you is that these organisms do interesting things during their life cycle, which is typically about 2 days, the way we grow them in the lab. And they're grown on a day-night cycle because they're plants. They're basically photosynthetic, so they need dark and light to do all the metabolism that they do. And if you pick it up in the morning of the first day, like in the book of Genesis, um, the daughter colonies that I show here as these smaller spheres hatch out of the mother. Sadly, the mother then dies. And now you have a small daughter colony which has its own germ cells. That's like the grandchildren of this mother. And during the first day of light, the germ cells grow very large. And then at night, they go through 9 or 10 rounds of binary division to produce 2 to the 9 or 2 to the 10 cells that will be the next generation. And interestingly, those new colonies, let's call them spheroids, have their germ cells on the outside, and the poles of the cells that have the flagella are facing inward. And that doesn't work because it's like me trying to swim with my arms inside. You have to turn yourself around. And so there's an amazing process called inversion or eversion, in which these spheroids literally open up and flip themselves around. I'll show you a video in a second, thus righting the wrong that was originally programmed developmentally. Now the flagella point out the germ cells are on the inside. And then the rest of the time, all that happens is the extracellular matrix expands and the whole colony grows without cell division. So we were happily studying this. We've written a number of papers on this inversion process using light sheet microscopy to show what's going on. So here is a video on the right showing the slow growth of the mother and then the sudden near synchronized inversion of these daughter colonies, not with high temporal resolution, but on the left, you see a close-up zoom of what an individual daughter colony does. And you'll see that there's some funny denting and deformation of this sphere, but then it develops a shape kind of like a mushroom. The anterior pushes through, the whole thing turns around, and it turns itself inside out. So this is eversion of a sphere done in biology. And it's a beautiful problem, and there's lots of interesting physics and math there as well as biology. But this is what we were doing when, basically in the middle of the pandemic, we got an email from a group in the US, Georgia Tech. This is the group headed by a biologist, Will Ratcliffe, and a physicist, Peter Juncker. And we got an email from Tom Day, who was a graduate student in the group. And he basically said, we've been studying an interesting multicellular organism called snowflake yeast. And I'll tell you what that is in a moment. And we've been looking at the geometry of its three-dimensional structure. And we're just wondering, do you by any chance have 3D image stacks of Volvox that we could also study our way and see if we find the same kind of behavior? And well, coincidentally, we did. So we teamed up with them, and I'm going to tell you what we found. But the first thing to say is, what is snowflake yeast? So in a textbook description of yeast, you will see it described as a unicellular organism that you grow up in liquid culture. So a group of biologists, Will Ratcliffe working with others and Mike Travisano in Minneapolis back in 2011, or published in 2012, wrote an amazingly important, beautiful, creative paper in which they created a new organism on Earth they called snowflake yeast. So the experiment consists of this. You take a tube of yeast growing in fluid, you centrifuge it so that things settle to the bottom, and you throw off the top, I don't know, 97% of what's there, and just take the bottom 3%, whatever settled fastest. Put that in a new solution, in a new tube, let that grow up, then do it again. And do this maybe 20 or 50 or 100 times, always centrifuging and selecting for whatever settled fastest. And clearly, the larger things are, the faster they're going to settle, if you just think of the settling of a sphere in Stokes flow. And so what they found after scores of such runs is that um, the yeast at the bottom of the chamber was no longer unicellular, but rather it was this ramified form you see at the bottom of these images. And it was not only just ramified and a cluster that might make you think it's just sticky particles adhering together, but actually when this multicellular object reproduces, the daughters, the progeny, are themselves multicellular, which proves that this has turned on essentially a hidden genetic version of the problem in which it's actually intrinsically multicellular. And they found it by selecting by gravitational settling for the ones that were multicellular. And it's a really interesting problem. There are many interesting things to say about it. But the particular issue they were focusing on when they contacted us was the geometry of these clusters. Now, Peter Juncker comes from a background in colloidal and granular physics, and he was thinking about this from using the language of Voronoi tessellations. So you have a ramified cluster like this, and you construct the lines connecting the centers of adjacent yeast cells. And then you look at the perpendicular bisecting planes of those lines, and the collection of those planes defines polyhedra around every cell. And they were investigating the distribution of the volumes of those polyhedra as kind of proxies for the space occupied by each cell. And they were asking us, can you do the equivalent for Volvox? So for that, we basically would look at the surface of Volvox, where all these cells are, and we just did a Voronoi tessellation using these points as the seeds, and we end up with Voronoi polygons in the plane. And we discovered something quite unusual and unexpected, which is that both in snowflake yeast and in Volvox, if you look at the probability distribution of the volumes associated with each cell, so that's a 3D volume for yeast and it's an area for Volvox cells, the probability as a function of the volume, here normalized by the volume of the small cell. It's a very broad, highly skewed distribution. It's not a Gaussian. It's a highly skewed distribution. And amazingly, both for snowflake yeast and for Volvox, it conforms to something known as a K-gamma distribution. So in red, I show here the particular form. So the probability of finding a volume or area, around a cell is basically x to the k minus 1 e to the minus kx, where x is the volume minus the cell volume divided by the mean volume minus the cell volume. So it's just a shifted normalized volume. So it's a K-gamma distribution, very broad, very skewed. And, Volvox appeared on Earth the better part of 1 billion years ago. Snowflake yeast appeared yesterday. They have utterly different reproductive processes, life cycles, et cetera. And yet, magically, they both have this kind of distribution. So where does this come from? So we spent a long time after we published this paper, which was just reporting the results, trying to figure out, is there some generic feature of life that might give rise to this behavior? And I'm just going to tell you the essence of what we think might be going on. So after a fashion, again, my student Anand Srinivasan, who comes from a highly mathematical probabilistic background, stumbled upon a whole literature that we knew nothing about in pure mathematics that turns out to be studying this exact problem. So these are Voronoi tessellations of what are called point processes. So basically, to summarize it in simple terms, you take a plane or a manifold and you throw down points according to some random processes. And then you basically make a Voronoi tessellation of the surface that you get from that, as you see over here on the right. And it turns out, to a very good approximation, quantitatively, numerically in 2D and 3D, the probability distribution of these areas is a gamma distribution, is a K-gamma distribution. It seems to be generic for tessellations of these kinds of random processes. So that, and I'll say a little more in a moment, but that kind of suggests that the reason it's generic is perhaps something about throwing down points within this extracellular matrix or in general in these biological structures. So here's a simple argument to see why this might have anything to do with the process. And you know the story probably about consider a spherical cow as a crude approximation to something. Let's consider a linear Volvox. So let's imagine throwing down points on a line where the spaces between the points represent the extracellular matrix. And just for a moment, consider a process in which the probability of creating a length of extracellular matrix around a cell is a Poisson process, so exponentially distributed with a rate constant lambda. So if x and y are the amount of extracellular matrix around two cells, both distributed this way, then to make a Voronoi tessellation, you have to do two steps. You have to take the sum of those two lengths, and then you have to divide by 2. So the first thing you learn in probability is that if you take the sum of two random numbers, the probability distribution is the convolution of their individual probabilities according to this. So if z is x plus y, the probability distribution of z is z e to the minus lambda z. And then you learn that if you multiply a random number by a constant, there's another rule for how you construct the probability. And so you discover that if you take the Voronoi tessellation of this one-dimensional problem, you get a K-gamma distribution with k equals 2. And the amazing thing is that Volvox has k equals 2.3. So it suggests, doesn't prove, but it suggests that there's something about the production of the extracellular matrix and this Voronoi construction that just naturally lands with K-gamma distributions. Now, the other thing you can do is you can think about dynamics where you produce this extracellular matrix in bursts, one burst or two bursts or many bursts, a few large bursts with a lot of material or many very small bursts, say all adding up to the same. And it's easy enough to show that you'll get k equals 2b for this bursty process. And if b is much greater than 1, you'll just get a Gaussian distribution by the central limit theorem. But is there any biological reason to suspect that the burst number is low? And it turns out there is precedent for this in studies of protein expression in bacteria, where some beautiful work dating back 20 years in which fluorescently labeled proteins that are produced end up binding to the membranes so they don't escape off into the world. So you can actually quantify the fluorescence and know how many proteins were produced. And in this particular system, you look at the probability of number of bursts per cell cycle, and it's actually got a mean value that's 1 or maybe a little bit more. And sure enough, the probability of distribution of molecules is exponential. So the basic argument we think might be going on here is just to appeal to the central dogma in biology. You have the gene, the DNA. It gets transcribed to form messenger RNA and then eventually to form proteins, which are exported. And if the degradation rate of the messenger RNA is really fast compared to the degradation rate of the proteins outside, you will get a single or a small number of bursts. And that might be what's actually explaining what's going on here. Now, I said, let's use Voronoi tessellations, but as I indicated earlier on in those images that Renzo was asking about, there is an identifiable boundary in these cell compartments. You see it with the microscopy, and actually with the group of Armin Hallman and his postdocs, Benny and Eva Vanderheide, husband and wife team, we've just published some work in which they have genetically engineered volvox to have a fluorescently labeled protein that lights up these compartment boundaries. So you don't have to do a Voronoi tessellation anymore. And you can actually see the boundaries as localizations of this particular protein. And then if you superimpose the somatic cells, you can see where everybody is. So you can actually quantify in enormous detail the real compartment boundaries. And the really amazing thing is that the Voronoi tessellations are remarkably close to the actual compartment boundaries. So it's like nature knows how to make a Voronoi tessellation, which raises all sorts of interesting questions about this self-organization process. Okay, so I'm continuing this kind of whirlwind tour of these three issues because I want to tie them all together. So let's talk about choanoflagellates now. So back in 2019, a group of biologists at the University of California in Berkeley, under Nicole King, a very distinguished biologist who studies choanoflagellates, reported the discovery of a new species of choanoflagellates called Choanica flexa. So here's the typical geometry of an individual cell of a choanoflagellate. It's a sort of ellipsoidal cell body and a single flagellum that it uses to swim and filter feed. And it's got a collar around it of little polymerized filaments that are called microvilli. It looks like a badminton shuttlecock with a flagellum. And they discovered a new species when they went on a field trip to an island in the Caribbean, as you do. And this species doesn't have an extracellular matrix, but it's a raft of cells held together by adhesive contacts between these microvilli tips. So think of it as just a, not quite flat, as I'll explain, raft of cells where every neighbor is connected to others by sticky contacts between these microvilli tips. And the amazing thing is that, as you can see here, I hope in this video, you can see the flagella beating, you can see some undulations of this structure, but there's not just one form of this organism. It actually flips back and forth between two different forms. In one, the cells are far apart, and it turns out the organism is a bowl-like structure with the flagella pointing in. So the cells are far apart, the flagella point in. It's not very good at swimming, but it's very good at filter feeding because of the porosity of that network. And the other form is a much more compact structure where the cells are very close together. And now this bowl has flipped to the opposite curvature, and the flagella point out. And this thing is very good at swimming and not very good at filter feeding. And the transition between these two is triggered by changes in light or by chemicals. So it's a really interesting sort of bistable elastic sheet. And we wondered how to describe this mechanically, but it has a lot of similarity to what we saw with the inversion of volvox. So as soon as they published this, we invited Thibaut Brunet and Ben Larson to come for a week to Cambridge to talk to them about this and try to brainstorm about what might be going on. And here you see other images of the two colonies in and out. And you can see the microvilli in this particular micrograph all meeting in these polygonal regions. And we now understand through their work that these collars that line the cell, the periphery, are actually encircled by a contractile ring of polymers and molecular motors. And when that ring contracts, it flares and changes the angle of this collar. And that manifests itself as a change in the curvature of the whole structure. And if you're clever with your image analysis, you can actually identify the polygonal structures formed by these adhering tips. So if you think about how you might make any kind of mathematical model of this, you have to think of a football, a soccer ball, right? Because if I'm going to make a curved structure out of packing cells together in some regular geometry, I'm going to need not just hexagons, but pentagons. So the way we started making a mathematical model of this was to try to locate the spheres that represent the cells as lying on the vertices of a geodesic icosahedron, which is just a projection of an icosahedron onto the surface of a sphere. And that naturally builds in pentagons and hexagons so that the system has curvature. And then we said, well, let's make a simple model of the connections between these cells by basically putting springs. So basically saying that every pair of neighbors is connected together by some number of elastic objects, which have maybe a spring controlling the angle that they make with each other, and then some elasticity for extension. And they're all connected together according to the geometry of this whole raft. And you can understand many things about this problem. You can, for instance, as you dial up the curvature from negative to positive and you flip this thing around, you can examine the swimming speed that you would get as a function of the swimming speed of a single cell by just associating a single point force acting on the fluid as a proxy for the flagellum. And you can discover, not surprisingly, that as you change the curvature, the swimming speed decreases when you go from the more porous state to the less porous state. Sorry, from the less porous state to the more porous state. But the most interesting thing is that those simple ingredients that I told you actually can allow you to understand the flipping of the curvature. So you just change the angle that these two meeting microvilli make with each other, and you can induce the system over time to flip from one curvature to another. So it kind of has this intrinsic bistability. And I have to say that as much as we thought this is great, we understand bistability, I couldn't quite figure out intuitively how we got this, let's call it very nonlinear property out of what is essentially just Hooke's law in the problem. I took a bunch of particles together, I hooked them together with linear springs, linear elastic springs and linear angular springs, and somehow out of that I get a bistable system. And I kept scratching my head before we submitted the paper. I really wanted us to have some intuitive understanding of this. And then I remembered a paper from long ago that I think I first read as a postdoc. It was by David Sung at Harvard and his then PhD student, Sebastian Sung, David Nelson and Sebastian Sung, sorry. Defects in flexible membranes with crystalline order. And this was a time that people were starting to worry about membranes of many types. And one of them was a polymerized membrane. Think of it just like a molecular model you would make with plastic model with spheres connected together by rods. But imagine that you introduce a pentagonal defect in this otherwise hexagonal pattern, or a heptagonal defect with 7 sides. And their diagrams here show what you can learn from everyday experience is that pentagons will tend to make curvature. This defect, this topological defect will make curvature so that you get a bump, but a heptagon will make a saddle shape. And I kept reading their paper over and over again and thinking somewhere there must be some idea of bistability, but I don't see it. And then suddenly I realized that when they at some point say in the paper, okay, let's imagine there is a defect and there's a perturbation of the shape up, let's say, that they could have equally said, let's make a perturbation down. And there's nothing in the physics of the system to distinguish up from down. So any result that they found that pertained to a bump up for a pentagon also pertained for a bump down. So there really is a bistability because there's no symmetry breaking in the system. So then I thought, oh, well, if this is right, then we ought to see this in some places. And the place to look first, actually, is this organism gonium I told you about before, the green algae with 16 cells. And I have looked at this image of gonium, which is from the web, a beautiful microscopy image, for a long time before I realized that actually, if you look at this cell, it actually has 7 neighbors, whereas the others have 6. So this actually has a heptagonal defect. And my then postdoc, Hélène de Malaprade, had been studying gonium in the lab, and here's a video of it, where if you look at it in the right moment, you can see actually that it formed a saddle shape. So indeed, a heptagonal defect makes for a saddle. And so after this, I said, okay, I just went out and ordered overnight online a molecular modeling kit, and I made a plastic model of mostly hexagons, and I put in some pentagons. And sure enough, it bends like this. And if you take this model and you push hard enough, you can flip it around and it curves the other way, and it's equally happy. So that's the origin of the bistability. And so then we went back to our colleagues and said, Brunei and Larson, and said, can you find all the pentagons from your confocal images of these organisms if you look in the right plane. So they found the right plane, color-coded everything by polygon type, and discovered that something on the order of 22% of the polygons in these systems are pentagons. So this is the fundamental reason why there's bistability in the problem. It's because of the presence of all these pentagons. Okay, so the last bit of story is going back to the Volvox extracellular matrix. So I showed you already this graph, this diagram of the life cycle, and I wanted to emphasize that after all this stuff happens on the inside with the daughter colonies, there's just an expansion of the extracellular matrix that enlarges the organism over time. And several years ago, my postdoc, Jane Chewy, started investigating the Volvox extracellular matrix by taking advantage of a mechanism to break up the colonies that involves hydrodynamic shear. So you take a suspension of Volvox and you mechanically shear it sufficiently, and it turns out that it breaks up into pieces. And here are some still images, and here's a video showing you, this is a high-speed video played back slowly, showing you that this object has rolled up into the form resembling a seed pod. So it's curled up on itself. It's still live. The flagella are still beating. It still turns around its axis, but it's all curled up in some very interesting way. And we've been wrestling with what is the origin of this, let's call it spontaneous curvature of these organisms. And after a fashion, we've come to understand that there probably are two effects going on here. One is that there are lots of topological defects in the packing of the cells in the extracellular matrix. And the second, there's something going on about how the matrix itself grows. But let me just show you the packing problem. So when we do light sheet imaging of these colonies, we can identify every cell in the entire colony. And from this, say, just make a Voronoi tessellation, we can identify the neighborhood of every cell. And we can color code that and count the number of pentagons, the number of hexagons, number of heptagons. And as you see here, for a cell with about 1,000, sorry, for a colony with about 1,000 cells, there's something like 200 pentagons, a slightly smaller number of heptagons. There always has to be an excess, that's a theorem, but obviously a sea of hexagons. And if this idea about these topological defects is valid as one possible explanation of what's going on here, then we ought to see bistability, at least sometimes, in the Volvox extracellular matrix. And here's a very striking image showing the streak lines of tracer particles around one of these pieces. And what you observe is that it's curved inward, like it would be from the original sphere, but all the motion is taking place on the inside, which means the flagella are on the inside and not on the outside, which means this piece has flipped around. So there's some evidence of bistability even in the Volvox extracellular matrix. But this is a complicated problem, and there's more to say about it. And what I'll just summarize for you is that we're now at the stage where we can take these pieces and from three-dimensional imaging actually extract their entire shape and start looking at their curvatures and other quantities and try to explore different possible explanations for what's going on. So here are some examples in the middle column showing you energy minimized shapes when we build these topological defects into an elastic theory of the membrane of the extracellular matrix. And here are the similar kinds of shapes of these finite pieces that come from looking at stretching energy within the extracellular matrix, which we think also plays a role. So somewhere in all of this, we think that the topological defects are playing an important role in organizing the 3D structure. Okay, so the last and very short thing I want to say has to do with sponges, and this is really just an idea. So I've shown you already that textbooks tells us there are three different main kinds of sponges, and the most interesting are these so-called leuconoid sponges with these spherical chambers of cells. Here's a beautiful electron micrograph by Sally Leys and collaborators. Sally is one of the world experts and leaders in the field of sponge biology. And if you look at this choanocyte chamber, it's full of these filter-feeding cells that look a lot like our choanoflagellates. And in fact, you can even find a pentagonal packing defect in there. So it's not surprising that we start to think that there's some interesting connection between the curvature found within these choanocyte chambers and the curvature we see in C. flexa, this bistable sheet. But the only way we're going to make progress on this is be able to look inside living sponges. And here's just a teaser of a video, a sequence of slices with confocal microscopy through a living sponge. done with our collaborators Kenji Kikuchi and Takuji Ishikawa in Japan, in which both a combination of microparticles that light up yellow that have been ingested by the sponge and dyes that label the nuclei or cell membrane. As you slice through this uh, object optically, you can see the choanocyte chambers and start to get a sense of how they're connected to each other and ultimately how they develop in time. So that's what I will hopefully report to you the next time I get invited, if I ever get invited back. Okay, so I claim, and here's a paper in progress where we actually achieve the truly shortest abstract in the world, which is, does biophysical research require a hypothesis? I say no, in the sense that I certainly didn't go into this thinking we were going to find K-gamma distributions. I had no idea about this. We had to just follow our nose and use our ideas from physics to try to understand how to make progress here. This is something I'm working on with Kyriakos Leptos, who is my longtime collaborator here, and Wilson Poon in Europe. But really, if I return to those referees who made me write that paper for eLife, one of the things I thought I would close with, again, trying to be a little mentorial here, is to consider some comments made by the reviewers of one of our papers. The paper was published, and so I have no qualms about this. But one of the reviewers said, the data should be described and the inferences drawn and the modeling, get this, relegated to its proper place as quantitative verification of the inferences that can be made directly from the data. And then later said, instead, the authors should let the data speak for itself and postpone heavier theoretical analysis for later, perhaps in the discussion. And that's the tension that we come across here, that there's different ideas of how to interpret data. But I would ask the following question. What language does the data speak if we're supposed to listen to it? And I claim that data says different things to different people, and it depends on your background. If you're a biologist, you see one thing. If you're a topologist, you see another. And probably if you're an applied mathematician, you see something else. So with that, I will stop. And I just want to dedicate this to the memory of my dear collaborator, John Kessler, who passed away several years ago and was a real motivation for us to get involved in this kind of work. Thank you.
Thank you so much thank you, Ray. It was a fascinating journey into the origin of life, if we may say, say something like that. I'm sure I have really lots of questions. I like to ask one first and then here. Sure. Is there any, of course, there are so many interesting things you talked about. Okay, one of them is just an easy aspect. Is there a relation? Can you establish a relation between the number of pentagons and the mean curvature or the Gaussian curvature of the surface? And then, given the fact, if I understood correctly, that these pentagons are kind of, or the length of the boundary in pentagon or hexagon, etc., is a way to interpret the amount of protein involved, or then is energetically expensive.
So I'm with you.
If you go for higher sides, higher polygonal sides.
So I would say two things. The first is that if you think about where did the pentagons come from in the first place, you have to think about the fact that the daughter colonies, let's say here, grow up inside the mother in some embryonic vesicle. And that's a packing problem itself. You have a cell that divides and divides and divides, and it fills this bowl. So the fact that it fills the bowl on the surface means it must have pentagons in the first place to smoothly do that. Otherwise, it would have all sorts of wrinkles and folds. So somehow the process of cell division under the constraint of being inside a sphere and lining the inside of the sphere is building in a bunch of pentagons in the first place. The cells are connected by little things called cytoplasmic bridges. And so the connectivity, the neighborhood connectivity, is set in even before there's any extracellular matrix. And that's built into the daughters from the beginning. So in some ways, the inversion process could actually be thought of as a bit of this bistability as well. So that's kind of hardwired, and you just have to live with that, I would say, if you're a Volvox. But for sure, of course, there's a huge amount of work. The Gaussian curvature can be determined or dictated, let's say, by the set of topological charges out there, the pentagons and heptagons. And but there's more to it than just that because there's also this elastic matrix that things live in. But if you think of the Gaussian curvature, of course, it's the product of the two curvatures, and so it doesn't distinguish one bowl curvature from the other. I would have changing the sign of both my principal curvatures will give me the same Gaussian curvature. So already, if you say what's determined by the defects is the Gaussian curvature, you already have bistability kind of built in. So then there's the separate question of does this have any implication for protein export and all of that? And we don't yet have enough information on a correlation between the pentagonal defects and their size, but we're working on it. And that might be interesting. Maybe it's true that the local neighborhood is stressed in some way, or maybe stressed less than others. It could be such that the protein production is different. But it's a good point. I hope that answers
Thank
your
you very
question.
much. I have to leave the floor to others. Other questions, please. Keith, go on.
Unmute yourself.
There is a question in the chat.
Oh, in the chat.
Can you hear me now? Yeah.
Yes.
Yes.
Well, my question relates to the bistability situation as well. If you consider a bistable situation, I think of a stable configuration as being a minimum energy state in some sense. So you have two minimum energy states. And I just wonder, Ray, if in the context you're considering, you can determine an energy for the two states involved.
Yeah. So I think so. I mean, going back to the work of Song and Nelson, it's kind of there, but not maybe in the most obvious formulation that there ought to be a kind of double well potential in the problem.
Yeah.
And the tricky thing is that you might think of it in terms of the mean curvature having a double well structure, but and in some ways, yes, it does, but it's really driven by the Gaussian curvature. So you have to kind of unfold the two branches of solution for saying the Gaussian curvature is something, and then I think you'll get a double well potential. But the fact that there's a barrier is clear because there's a huge amount of stretching energy if you were to keep it flat. So you relieve all that by buckling because then you allow for the curvature that it wants. But to keep it flat when it's got pentagons is just a lot of stretching energy. So there's clearly a barrier. So I'd love to be able to see some double well potential. Yeah, that would be great. I could read out sorry, if you finished or no?
Well, I was very interested in this because Renzo may remember, and Xin Liu also, a couple of years ago, I was trying to figure out another bistable situation involving topology. This is the very simple situation of a knotted configuration. If you take a knot like the trefoil knot, there are two possible fairly elementary configurations, the T2-3 or the T3-2 as it's called. And they have different energies and there's a barrier between them. And your discussion sort of reminded me of that. You've got to get from one to the other. You've got to really push it over the barrier. There's got to be some force involved.
Yes,
and
yes.
I suppose that's the case with you also.
Yeah, and in fact, I think perhaps the simplest system to study here would be the C-flexa one. Although there are some Volvocalyes like Gonium, which, sorry, not some others, larger ones, which actually undergo a kind of wimpy flip, a little wimpy inversion, where they change their bowl-like configuration. But C-flex is the best. And what we're trying to do now is actually get high-speed imaging to watch this interconversion. And what is the geometry? Is it, there's some sort of barrier crossing, but does it proceed by some little region that locally flips and then it spreads, or is it something more global? It's a little bit like putting your socks on and rolling them up and unrolling them. It's a kind of there's an issue there. And it turns out from the work of Hallman many years ago, we know that as you consider larger and larger examples of the Volvocalyes, there's an ever more elaborate folding that occurs during inversion. So there is something systematic from the elasticity theory that really speaks to this bistability. And that's what we're trying to figure out.
Wonderful.
So I noticed someone said there was a question in chat, which I
Yes,
found. Should I read it out?
can,
I can read it
can,
out, or you read it out?
yeah, I can read it out for everybody. Marian
Okay.
Taya says, could chirality be considered a major driving force in the origin of multicellular life? And how external physical forces, such as magnetic fields, polarized UV light from the sun, could influence how cells make their structures?
So, excellent question. I would say that when you look at all of these organisms in this lineage, I'm not going to try to speak globally about all of life, but uh, one of the interesting things you see is the chirality across scales. So an individual Chlamydomonas, when it swims with a breaststroke, but the two flagella twist slightly as they beat so that it actually spins around the axis that points in the direction in which it moves. So it spins like a rifled bullet. And every one of these species, in one way or another, translates and rotates around a body fixed axis. So gonium, for instance, as you saw, it's a plate that as it moves, say, this way, it actually rotates. And there's a complicated arrangement of flagella that make that happen. Every one of these organisms rotates and spins. And it's also true that every one of them is photosynthetic and phototactic. And although we don't know the chicken and egg answer here, we know that rotating is crucial to be able to do phototaxis. Now, whether that came first or second, I don't know. But the process of swimming toward the light and turning toward the light is intimately connected with the spinning motion. So you would be okay, probably, to imagine that there is a connection between chirality and multicellularity. Whether it's causal, I don't know. We've looked at it from the point of view of how does this one do phototaxis? How does gonium do phototaxis? How does Volvox do phototaxis? And we did find, this would be another talk, we did find an interesting conserved quantity, which is a tuning between the spinning frequency and certain timescales associated with the flagellar motion. And that tuning holds over 3 orders of magnitude in cell number. So I would say more like it allows multicellularity, or it's preserved by multicellularity, but I wouldn't say it's required because there are other types of organisms that don't do this. But chirality in general is present all over the place. It might not be spinning of the organisms, it might just be chirality of proteins or protein structures. I hope that answers your question.
Okay, thanks. Other questions, please. I have a rather philosophical kind of comment, question. We can see it throughout your many levels, from the very primordial cell types, a kind of game that is, on one hand, optimizing shape, energy, etc. On the other, a bifurcation in symmetry. You grow preserving symmetry somehow, which to me seems to say I economize as much as I can, I optimize as much as I can the energy I have, and then something new must happen, maybe in terms of reproduction or other reason that breaks this context, and you gain a new shape or new activity. What is your view, Ray?
I agree that that's a very useful way to think about the problem. You can think about, and we've done this some years ago, you can think about the scaling laws that would hold if you were to continue to grow as a sphere, arbitrarily large. So imagine that your building blocks are always these individual cells. And maybe in the simplest case, you just imagine the density of cells on the surface of a sphere is constant, but you just consider larger spheres by saying larger number of cells. And then just ask, what would be the swimming speed? What would be the nutrient uptake under simple laws of diffusion or advection and things like this? And you readily realize that there are a whole bunch of interesting things that happen as you get larger. So you will tend to sink faster. And you will tend, just like the snowflake yeast problem, you may swim faster if you actuate your flagella, but if you were to turn your flagella off, you will sink faster. You could also ask about nutrient uptake, and what you discover is if you're a sphere, the rate of uptake by diffusion will just be proportional to the radius. That's a standard Smoluchowski law. But if you have all your tissue on the surface of the sphere, your needs scale like the square of the radius because it's the surface area. So as you get larger, you won't be able to keep up with your needs because your needs will grow like R squared, your intake will grow like R. On the other hand, these organisms swim and they create a flow, which actually turns out to be a high Peclet number flow. So actually, it's advection diffusion that matters when you get to larger organisms. And if you think about the advection diffusion problem for these things, it turns out the Peclet number is large enough that you would end up with boundary layers that enhance your diffusion and actually make the uptake rate go like R squared. So that says, it's good to have flagella because then you can get bigger. But then the problem is, and this is a piece of biology, that when cells, like bell bugs, go through cell division, they lose their flagella transiently because the microtubules that make up the flagella are actually used inside the cell to pull chromosomes apart during cell division. So this is called the flagellation constraint. You can't multitask. You sync or divide, but you can't do both at once. So imagine that you're a very large sphere. You're beating your flagella. You get all the uptake you need, but then you go through reproduction. You turn it all off and you start sinking. That's not going to be good because reproduction takes a while. And the bigger you are, right? So it seems one way to answer the question of why there are two cell types is to realize that if you sequester the reproduction on the inside in specialized cells and let the galley slaves keep doing their thing, then you get all the nutrient uptake that you need, and you still go through reproduction and don't encounter this problem. So it can be an argument along the lines of the biology, the biological fact of this flagellation constraint drives the division of labor beyond a certain point. And you can kind of back this up with some calculations. So that's one answer to where the symmetry rate can come from. It's actually a particular biological detail. It's not just some imagined, Higgs boson. Right? There's a particular issue that the cells have to deal with.
Very interesting. Thank you. Other questions? Okay. If not, well, I thank you on behalf of everybody. Thank you so much, Ray. Wonderful, wonderful talk, I have to say.
Thank you, Renzo, for the introduction, for the invitation. Sorry, for the invitation. And thank you all for coming. Pleasure to meet you and talk to you.